Hello students, we are going to start with lecture 2 of lesson number 1, that is reproduction in lower and higher plants. Last topic we have studied was different types of pollination. Now we are going to start with outbreeding devices. Here is a chart which shows different types of outbreeding devices: unisexuality, dichogamy, prepotency, heteromorphy, hercogamy, self-sterility. From these different types, dichogamy is further having two types, that is protandry and protogyny. Students, many plants have mechanisms that prevent self-pollination. Now we know that in self-pollination, variation or genetic diversity cannot be seen. So to promote cross-pollination and increase genetic diversity, plants they have naturally evolved wide varieties of sexual strategies. Genetic diversity is very essential for natural selection or evolution which is carried out by natural selection. Thus, plants have developed many devices to encourage cross-pollination. So, these following are some examples of the outbreeding devices. So, first one is unisexuality. Now, the plants bears either male or female flowers. It is also called as dioecium. Self-pollination is not possible in this flower. Monoecious, for example, maize; dioecious, for example, mulberry and papaya. They show unisexuality. Dichogamy. Flowers are bisexual, but anthers and stigma mature at different times. There are two types of dichogamy: protandry, that is androecium become mature earlier than gynoecium, for example in sunflower, and in protogyny, gynoecium matures earlier than androecium, for example gloriosa. So protandry and protogyny, in this exactly two opposite mechanisms we can see, but both the types are of dichogamy. The next type is heterostyly or heteromorphy. Stigmas and anthers are placed at different levels, and then this prevents the pollen from reaching the stigma and pollinating it. So self-pollination can be avoided in this type of or because of this mechanism, that is heterostyly. For example, primrose. The next type is hercogamy. It prevents self-pollination in a bisexual flower. In plants, natural physical barrier is present between two sex organs and avoid contact of pollen with stigma of the same flower. So, self-pollination is avoided or prevented. For example, calotropis, or commonly it is called as rui plant. The next is prepotency. Pollen grains of other flowers germinate rapidly over the stigma. Then the pollen grains from the same flower, for example apple. Self-incompatibility. Germination of pollen grain on stigma of the same flower is inhibited. That is, to avoid self-pollination, pollen grain germination is not carried out in self-incompatibility. For example, tobacco plant. So students, this topic is also very important, outbreeding devices. So it may come to enlist outbreeding devices, or enlist and describe also outbreeding devices may come for 2 marks. So please go through the topic properly. Now next we are going to study about pollen-pistil interaction. Pollen-pistil interaction is nothing but the interaction of pollen grains with stigma. It begins with pollination and ends with fertilization. That means it starts with pollination and the final result is fertilization. The events from deposition of pollen grain on stigma, that is pollen grain are transferred on stigma, to the entry of pollen tube in the ovule are referred as pollen-pistil interaction. This is a definition of pollen-pistil interaction. That means the events which take place from the entry of pollen grains on the stigma up to entry of pollen tube in the ovule. So all these events are are called as pollen-pistil interaction. So in this, during pollination, numerous pollen grains land on the surface of stigma. The pistil has ability to recognize the right pollen of the same species and discard the wrong type of pollens. That means many pollen grains or from various pollen grains, stigma selects only desirable pollen grain for the pollination. This is carried out by special type of protein. Right pollen grain absorbs water and nutrient from the surface of stigma and germinates and produces pollen tube. So formation of pollen tube, this is nothing but germination of pollen grain on the stigma. So pollen tube moves through the style and reaches the embryo sac or up to the ovule. The tip of pollen tube enters one of the synergid, then ruptures it and release two male gametes. Here you can see in the nearby picture entry of the pollen tube in the embryo sac. We have studied the structure of anatropous ovule in detail. So in the embryo sac you can see there are different 8 types of nuclei. Next is artificial hybridization. It is a type of, or it is one type of the crop improvement technique. Desired pollen grains are hand pollinated and used for pollination or used for fertilization. The procedure used is emasculation and bagging. We are going to study practically about the techniques of artificial hybridization, that is emasculation, bagging and tagging. So emasculation is nothing but removal of anther to avoid the self-pollination. The next is entry of pollen tube into the ovule. Pollen tube germinates or pollen tube penetrates the embryo sac by different pathways. Very first is porogamy. Pollen tube enters ovules through micropyle. We have studied in the VS of anatropous ovule that micropyle is a very narrow opening which is present at the apex. So when pollen tube enters through that micropyle, that is called as porogamy. The next is chalazogamy. Pollen tube enters ovules through chalaza. We have studied that chalaza is opposite to the micropyle, that is not very narrow opening. Then mesogamy. Pollen tube enters ovules through integuments. You can see here 3 diagrams are shown. The first one is porogamy. Here pollen tube enters through the micropyle. Chalazogamy. Here you can see entry of pollen tube through chalaza. And here in the third diagram, that is mesogamy, pollen tube enters through the integument. Let us now study about very important topic, that is double fertilization. Here pollen tube enters the ovule through the micropylar end. We have studied the 3 types of fertilization or 3 types of entry of pollen tube. So when that pollen tube enters the ovule through the micropylar end, it carries 2 male gametes and penetrates one of the synergids. The synergids ruptures and releases the 2 non-motile male gametes in embryo sac. As non-motile male gametes are carried through hollow pollen tube, it is known as siphonogamy. You can see here in the picture how germination of pollen grain takes place. Pollen tube enters through the micropylar end and then it reaches the egg cell by breaking of synergid. So when synergid breaks, 2 male gametes, they enter into the embryo sac. Now here as 2 male gametes are present, first male gamete fuse with the egg cell. The fusion is called as syngamy. It forms diploid zygote which develops into embryo. Second male gamete fuses with secondary nucleus to form endosperm or primary endosperm nucleus. The fusion is called as triple fusion. Primary endosperm nucleus develops into triploid endosperm. Here in this process, fertilization takes place twice and therefore this process is called as double fertilization. in which one male gamete fuses with the female gamete, second male gamete fuses with the secondary nucleus, and therefore formation of zygote and formation of endosperm take place, and therefore this particular procedure is called as double fertilization, as you can see here in the picture. Here are the significance of double fertilization. Fertilization stores food material in seed. Diploid zygote develops into an embryo. The triploid PEN develops into nutritive endosperm tissue. It restores the diploid condition by fusion of haploid male gamete with haploid female gamete through syngamy. It also helps to avoid polyembryony. Polyembryony means development of multiple embryos. Let us now discuss about development of endosperm. Nuclear endosperm. Primary endosperm nucleus repeatedly divides mitotically without wall formation to produce large number of free nuclei. A big central vacuole appears in the center of cell, pushing the nuclei towards the periphery. Later on, it forms multicellular endosperm by cell wall formation. In certain cases, cell wall formation remain incomplete. For example, wheat, sunflower, coconut, etc. You can see here in the nearby picture. This is a picture of nuclear endosperm. Cellular endosperm. In some plants, division of triploid primary endospermic nucleus is followed by wall formation, so that the endosperm is cellular right from the beginning. For example, balsam, petunia and adoxa, etc. Here is a picture of petunia plant. So this particular diagram shows cellular endosperm. Halobial endosperm. It is intermediate between cellular and nuclear type. First division of primary endosperm nucleus is followed by a transverse wall which divides the cell uniquely. The smaller cell is called chalazal cell and larger cell is called micropylar cell. Then the nuclei in each cell divide by free nuclear divisions, and then walls develop between nuclei in micropylar chamber. For example, asphodelus. Development of embryo. The development of zygote into embryo is called embryogenesis. That means zygote when converts into embryo, that is called as embryogenesis. Here are some stages how zygote develops into mature embryo. So this takes place in the dicot embryo, or this is a dicot embryo. Here is a tree diagram which shows embryogenesis. So the zygote divides to form two-celled proembryo. The larger cell is towards the micropylar end, that is called as suspensor initial cell, and the smaller cell towards the chalaza is called as embryonal initial cell. Now the suspensor initial cell divides to form 6 to 10 filamentous suspensor cells. The first cell forms haustorium. Lowermost cell, it is called as hypophysis. And embryonal initial cell, it gives rise to 8-cell octant. Now this 8-cell octant is further divided into several planes which are heart shaped. Then here this 8-celled octant, it shows lower tier form hypocotyle and radical, while upper tier forms plumule and cotyledon. So lower tier is forming hypocotyle and radical, and upper tier it forms plumule and cotyledon. Now this divided heart-shaped octant giving rise to two lateral cotyledon and terminal plumule. Now further after that, enlargement of hypocotyle and cotyledons result in a curvature of embryo and it appears horseshoe shaped. Please go through the chart once again. The process of entire embryogenesis is explained in this chart. So practice it properly. Here are the stages of development of monocot embryo. You can just go through the picture. In monocot embryo, single cotyledon occupy terminal position and plumule is lateral. Shield-shaped cotyledon is called scutellum. The protective covering of plumule is coleoptile and radical is coleorhiza. Let us now discuss about seed and fruit development. One of the ways that plants can produce offsprings is by forming or by making seeds. Ovules develop into seed, integuments develops into seed coat. Seeds have two coverings, testa and tegmen. Ovary develops into fruit. The covering of fruit is called pericarp. You can see here the picture of fruit or the vertical section of fruit. Pericarp, it is having 3 layers. Innermost is endocarp, middle is mesocarp and outermost is exocarp. Seed is also having 3 layers: endosperm, embryo and seed coat. Endospermic seed is also called as albuminous seed. In some seeds, the food reserves in the endosperm are partially used up by the development of an embryo. In such seeds, the endosperm remains conspicuous and fills a greater part of the seed. Thus, the resultant seed is endospermic or albuminous seed. For example, maize, coconut. Non-endospermic or ex-albuminous seed. In some seeds, embryo absorbs food reserve from the endosperm completely during its developmental stages. Thus, endosperm disappears in mature seeds. For example, pea, bean, etc. Significance of fruit and seed formation. Fruits provide nourishment to the developing seeds. Fruits protect the seeds in immature condition. Seeds serve as important propagating organs of plant. Seeds and fruits develop special devices for their dispersal and thus help in the distribution of the species. Seed dormancy. Mature and viable seeds are not germinate even through favorable condition is present. Dormancy is very important for the dispersal of seed. Dormancy, it is a metabolic arrest that facilitates the survival of organisms during adverse environmental condition, that is during unfavorable environmental condition. Structural or physiological adaptive mechanism for survival of plant is called dormancy. Lupinus articus 10,000 years, Phoenix 2,000 years. Next is apomixis. Apo means away and mixis means mixing. Formation of embryo through asexual method of reproduction without formation of gametes and fertilization. So this apomixis, it is exactly opposite of amphimixis. In amphimixis, union of male and female gamete is formed, while in apomixis, no union of or no formation of male and female gametes. So it is formation of embryo without formation of gametes. When a gametophyte cell produces embryo like structure without fertilization, it is termed as apogamy. Diploid sporophyte cell produces a diploid gametophyte without undergoing meiosis is called apospory. For example, orange, mango, etc. Here are the categories of apomixis. Recurrent apomixis. The embryo sac generally rise either from an archesporal cell or from some other part of the nucellus. In diplospory, the unreduced embryo sac is derived from the diploid megaspore mother cell. In apospory, the nucellar cells give rise to apomictic embryo sac. Non-recurrent, megaspore mother cell undergoes usual meiotic division and a haploid embryo sac is formed. Embryo arises either from the egg by parthenogenesis or from some other haploid cells of gametophyte through apogamy. Plants produced by this method are generally sterile and do not reproduce sexually. Nicotiana is a common example of the non-recurrent apomixis. The next is adventive embryony. Embryos may develop from somatic nucellus or integuments along with normal zygotic embryo. It gives rise to condition called polyembryony. For example, mango, orange, etc. The next is parthenocarpy. It is a condition in which fruit is developed without the process of fertilization. So here normal union of male and female gamete does not take place, no fertilization, no zygote formation. So ovary produces auxin IAA, that is indole 3 acetic acid, which is responsible for enlargement of ovary into fruit. The fruit resembles the normally produced fruit, but it is seedless. For example, pineapple, banana, papaya, etc. The last topic of the lesson is polyembryony. It is the occurrence of more than one embryo in a seed. It is of two types. Adventive polyembryony. An embryo develops directly from the diploid cell of nucellus and integuments, for example citrus. And cleavage embryony or cleavage polyembryony. Zygote proembryo sometimes divide into many parts or units, and each unit develops into different embryos, for example pinus. So here we have finished with the lesson number 1, reproduction in lower and higher plants. Students, study the different topics, then practice all the diagrams properly. There are some important questions like describe the process of double fertilization, explain the stages involved in the development of the gametophyte, male and female gametophyte. Then there are some short questions like describe 3 devices by which cross pollination takes place. Then there are some pollinating agents. So what are the agents of pollination? Okay. Then define megasporogenesis, define parthenocarpy, define polyembryony, define apomixis, define dormancy. These all definitions are also very important. There are certain diagrams like double fertilization diagram, then diagram of the anatropous ovule, vertical section of anatropous ovule, development of male gametophyte, then TS of anther, draw and label TS of anther, then draw and label complete flower or different parts of the flower. So students, practice all these diagrams and study well. All the best.